In the seminar we are presenting today, we are going to see a practical approach for the genetic construction of an adenovirus that carries the S gene from SARS-CoV-2 coronavirus. The resultant recombinant adenovirus will be replication-defective and could be used as a vectorized vaccine for COVID-19. In resume, we can say that in this seminar we'll show you to employ real genetic data to get a virtual COVID-19 vaccine. In this practical approach, we are going to use the tool US Cloning. This is a free application developed at the University of Santiago de Compostela in Spain that is aimed to be used by molecular biology, genetic engineering, or in general by biotechnology students to prepare their genetic in silico designs. The other tool we are going to use in our project will be the AD system from Agilent Technologies. All the information about this system and to carry out the project in the lab can be found in the catalog 240,009 from Agilent Technologies. We have chosen the ADS system because it's a versatile tool for delivery and expression of genes in adenovirus. As we will see in a moment, the cloning process is quite simple. Can be carried out in Escherichia coli, and finally the gene will be transferred from Escherichia coli to the adenovirus by homologous recombination. All things is done in Escherichia coli. The AD system we are going to use in our project consists in two vectors. pDC1 is an Escherichia coli vector because it contains the replication origin for this species and the classical ampicillin-resistant gene for selection of recombinants. Besides these regions coming from Escherichia coli, the vector contains the entire genome of the AD5, the serotype 5 of adenovirus, human adenovirus, except for two genes, E1 and E3. E1 is an essential gene for replication. Deletion of this gene renders replication-defective viruses. These viruses can only replicate within packaging cells that carry the deleted gene. On the other hand, E3 is a dispensable gene. It participates in evading host immunity. It's dispensable, and deletion of these genes creates room for up to 7 kilobase, about 7 kilobase of foreign DNA. More than enough for the S gene from the coronavirus. pADC1 is quite large. Its size is more than 30 kilobases, making it difficult to insert any genes directly in this vector. See the left arm and the right arm present in this vector. These regions are shared by this vector and the second vector of the system. The second vector of the system, pShuttle-CMV. Beside these homologous regions, the vector is designed especially for cloning of genes that have no promoter nor terminator of the transcription. pShuttle vector has an early gene promoter from the cytomegalovirus and the transcription terminator, in this case from another virus, SV40. The vector contains a polylinker region that is very useful to insert any gene using any of these enzymes. Our genetic in silico design will be carried out in 4 steps. In the first step, we have to get the S gene from the SARS-CoV-2 coronavirus. We will use the PCR technique to amplify the S gene with appropriate primers, that is, the introduction of the gene into the pShuttle-CMV vector under the control of the cytomegalovirus promoter. For that purpose, we are going to create a couple of primers for the PCR reaction, primers that will be flanked by sequences recognized by two different restriction enzymes, KpnI and XhoI. In the second step, we will clone the S gene into the pShuttle vector, just here. This will be done by directional insertion of the S gene at the KpnI XhoI sites present in the polylinker region. Here, this insertion places the S gene under the control of the cytomegalovirus promoter, ending the transcription at the SV40 terminator. In the third step, we will transfer the coronavirus S gene from the pShuttle-CMV to the adenovirus PAD-C1. This will be done by homologous recombination. Homologous recombination will be facilitated if we previously linearize the recombinant pShuttle vector, linearization that can be carried out by digestion with pME1. The homologous recombination will produce a circular vector that contains the AD5 sequence with the S gene from coronavirus, but contains additionally a sequence with the kanamycin-resistant genes and the origin of replication of Escherichia coli, this here. In the fourth step, we have to get rid of the region that contains the origin of replication and the kanamycin-resistant gene. This will be done by digestion with PAC1, and the virus resultant can be packaged with a cell line, special cell line, that will produce mature viral particles defective in replication. These viral particles released by the packaging cells could be used as vectorized vaccine for the coronavirus spike protein. The viral particles are not replicative, they are defective in replication, but can infect cells. Cells can express the spike protein, the body produces antibodies against the spike protein, and we will be protected for the infection of SARS-CoV-2 coronavirus. Let's start then our project. Open the application, click in Webs. As a preliminary step, we have to download the sequences from EMBL and from AdGene. EMBL, we search for SARS-CoV-2 complete genome, and we can download any of these sequences. This one, for instance. Download the EMBL format. Here it is. Close the web, open AdGene, search for PAD-C1, here it is, PAD-C1. View all sequences and download the gene bank format. Here it comes. repeat the operation with pShuttle-CMV. Search in plasmids. Here it is. Click View all sequences and download the gene bank format. Here it comes. Close, and now open the sequences. This is PADC1. Edit PADC1 and save with the name PADC1. Open the second. It must be this one. Here it is. Edit pShuttle-CMV. Save it. And keep it. Open then the one from EMBL. Edit SARS-CoV-2. OK, and save it. Now I'm going to close the application. See the sequences I have downloaded, these 3 sequences, and we have to create a new folder for our project. Create a new folder here in vaccines, and we are going to create a new folder, COVID-19. OK, we take the sequences and we are going to number the sequences because we are going to use a rational system, numerical system, for all our files. Here it is. The second step will be complete the maps with the data provided by Agilent Technologies and the catalog 240,009. In this catalog, we can see that the inverted terminal repeat of pShuttle-CMV starts in the base 1. Here starts in the base 440. Then we have to rotate the molecule so that the features match. Now starts just in the nucleotide number 1. Now we have to add 2 important features, the AD right arm and the AD left arm, because we need these features for the recombination process. We can edit these features and add these features. AD5R. Add and update. Here it is. We have to do the same with the left arm. Not to waste time, I've already done the job, and now we have the 3 sequences ready. As you can see, I've renamed some of these features, and I have colored some of them too, and I have done the same with the other 2 files. Well, the first step in our project is to amplify the S gene from the SARS-CoV-2 genome. Then, before proceeding, we have to check that the S gene encoding the spike protein has no sites for any of the enzymes we have to use in our project. Let's go. See that there is no site for KpnI. We add to the map, show 1, pME1, and PAC1. In fact, there are sites in the genome, but any of these sites lies on the S gene. Then we can employ these enzymes in our project. To carry out the amplification process, we have to create a couple of primers from the open reading frame of the S gene that encodes the spike protein. We view the open reading frame on the sequence. This application only shows the positive strand, and from this sequence we have to create the couple of primers. Remember, being this the open reading frame, the forward primer, we have to construct the forward primer from this sequence and the reverse primer from this. But the sequences must be written always from 5'N to the 3'N. Open the library, primers motif. You can add your primers here, or you can create a new file for our project, COVID-19. We have to add the primers here from the open reading frame. Then from this region, select about 40-50 nucleotides. Copy these nucleotides, name the primer, forward primer, and adjust the melting temperature of the primer about 55 degrees. Okay, that's right. Add the primer and go again to the open reading frame . Add here, but you have to create the complement and reverse. Complement and reverse. Now adjust the temperature. Okay, that's right. And add the primer. Update. And you have to add now a sequence for the 2 enzymes we are going to use in our project. KpnI for the former primer and XhoI for the reverse primer. We go to the restriction enzymes, list manager, and we see the sequence of KpnI. We have to add the sequence to the 5' end and a couple of additional bases. We have now a tail of 6 more 2 additional bases, 8 nucleotides. We can add the primer. Well, is this one, KpnI, and update again. And you have to do the same with the reverse primer. We have to add here the sequence for SHOW1. A couple of additional nucleotides. Adjust the tail. SHOW1. You can add a command, primer reverse for S-gene, etc. and update. Okay, we've got our primers created. This is unnecessary. This is unnecessary. Update. And now you have to map the primers here they are. We can carry out now the PCR reaction. Forward primer. Reverse primer. Okay. See the PCR product you have taken. In the description you can see the primers that you have used in this process, the melting temperature, information, interesting information. Edit the name and we save our file. Okay, here it is. To prepare the gene for the ligation, you have now to digest with the enzymes here and here. Now you've got your insert prepared for ligation. See that the PCR construct contains 2 sticky ends for KpnI and for SHOW1 here. Here are SHOW2. Save this molecule and in the description you can see the history of these treatments. The next step in our project will be to clone the S-gene into the polygator region of the shuttle vector pShuttle-CMV. Open the vector. The polylinker region must be between the C2-megalovirus promoter and the SV40 terminator, between the bases 906 and 11 and 17. Go then. Have a restriction map of the enzymes that just cut once, or better, take only a map of the sequence you are interested. Okay. As As you can see, there is a site for XhoI and another site for KpnI, the enzyme we are going to use to clone the SG. The next step will be to cut just with these enzymes, cut with XhoI and cut with KpnI. Here is the vector linearized, just ready to make the ligation with the insert. Save the molecule. 2.1 pShuttle-CMV cut with KpnI and XhoI. Save it and proceed to ligate the two molecules. For that, you have to load both molecules as you can see. The molecules have sticky ends for both enzymes. Ligate, and you have the S-gene inserted into the pShuttle vector. This is a classical directional cloning because the S-gene is inserted into the pShuttle vector in an unique possible orientation. The S-gene will be expressed under the control of the cytomegalovirus promoter, and the transcription will end at the SV40 terminator. The efficiency of the expression of the S-gene will depend on the quality of the form COSAS sequence in our construct. You can see here the canonical COSAS sequence and the COSAS sequence generated in our construct is a very good COSAS sequence, the best possible in our case. This is a powerful reason for use KpnI preceding the ATG start codon, because KpnI provides the ACC basis of the COSAS sequence. Coming back to the recombinant molecule, you can edit the name and add the enzymes you are going to use in the next step. You can add to the site for EcoR1 and see that the pME and EcoR1 just cut between the ARBs, the adenovirus ARB, linearizing the molecule. EcoR1 has another site in the S-gene and cannot be used for this purpose. To linearize the molecule, you have to use pME1. If you see the nodes, the application has generated a node, an automatically node that explains the ligation process. You can add additional information as the method of selection of recombinants or the way of distinguishing the recombinant plasmid from the original one. Like this. Well, save the molecule. And finally, to complete the second step, digest the recombinant plasmid with pME1. This digestion linearizes the molecule, freeing the adenovirus arms, and this facilitates the homologous recombination process. Edit the name and then save the molecule. Okay. In the next step, we have to transfer the S gene from this linearized molecule to the adenovirus vector PADC1. This will be done using the powerful Escherichia coli recombination system. And in this application, we have first to create the appropriate recombination sites. To create the recombination sites that direct the homologous recombination between pShuttle and PADC, go to Libraries, open recombination sites, select here homologous, new site, and from the ADR feature, select about, in the middle of the sequence, about 50 nucleotides. Copy and paste it here. Add the name AD5L. You can add any comment to save time. I am not doing that, and add the sequence. Repeat the operation to create the 85R. At the site and update. You don't have to create a acceptor site because in homologous recombination the donor and acceptor sequences are the same. Update. Okay, here they are, 85R and 85R. Now, to proceed with the recombination, go to the cloning menu, click on recombination, and load the second molecule, PADC1. Okay, select then homologous, intermolecular recombination, double, 85R, 85R, rec, and you will get two recombinants in this process, rec1 and rec2. Remember, you have to change the names of these recombinants if you want to keep them. Third, this is one of the recombinants. As can you see, the process of recombination took place here in the middle of the sequence. Since we have colored the sequence in different colors, here are the different colors of the new sequence. As you can see in this map, this is a linear recombinant that contains the origin of replication for Escherichia coli and the ampicillin-resistant gene. You can add this information to the notes, and being this a linear molecule, it cannot replicate in Escherichia coli and it will be lost in a few generations. Save this file and open the second recombinant. As you can see in this map, this is a circular recombinant that contains the entire genome of adenovirus, except for the genes E1 and E3. It contains too the S gene encoding the spike protein from coronavirus and a region of Escherichia coli. The region contains the origin of replication and the kanamycin-resistant gene. You can complete your notes with some of this information. You can add to the sites for the enzyme PAC1 these are the sites, and you can edit the name if you like. And save the file. Well, let's have a look in this region. As you can see, PAC1 just cut between the inverted terminal repeats of the adenovirus. You can get rid of this region just cutting with PAC1. Cut and cut. Select the largest fragment, and in this linear sequence we can see that the spike protein is expressed from the S gene, the expression from the cytomegalovirus promoter, and in the SV40 terminator. Digestion with PAC1 not only removes the region that contains the Escherichia coli replication origin and the kanamycin-resistant gene, but also renders a linearized molecule. This molecule contains the inverted terminal repeats of the adenovirus, flanking all the DNA, and contains the psi signal necessary for packaging. Then this means that this molecule can be used to transform a packaging cell line that provides the E1 protein needed to replicate the recombinant virus, and that this virus cannot synthesize. Add this information to the map, and as you can see here, the mature virions can infect target cells, can express the spike protein from SARS-CoV-2, but cannot replicate within cells. That means that after appropriate immunization testing, mature recombinant virions could be used directly as a vaccine for SARS-CoV-2. Edit the name and save your molecule. PAC1. Okay. Well, as you can see, at the end of the job you will have a folder that contains all the files that you have created. The files will be ordered numerically, and the number of each file indicate the origin of the molecule. Thus, the number 1.1.1 indicates that this molecule comes from the number 1.1, and in turn these come from the molecule 1. On the other hand, new numbers indicate new molecules. 1, 2 and 3 are the numbers of the starting molecules. 4 is a new molecule created in a ligation reaction. And 5,1 and 5,2 are two molecules obtained in the homologous recombination process. All the files correspond to the molecules you will have to create in the lab. Well, that is all for today. Sorry for my English and good luck with your lab work.